Hızlan, can you hear me? Could you start sharing the, or maybe you can share later. Now I will, I'm Masaru Ueno. I belong to biomedical program in Hiroshima University. Today I want to start 68th HiHA webinar, and today's speaker is Hızlan Ağuş, Istanbul Yüzyıl University, Turkey. And I will briefly introduce about his background. He got a PhD at Hacettepe University in Turkey, 2014. His major is molecular biology, and he got assistant professor position in 2015 at Current University. Hızlan and I have a similar strategy about research. We use fission yeast as a model organism to apply our result to anti-cancer drug discovery or as a cure of disease. We didn't have any interaction, but a graduate student in my lab asked Hızlan about some experimental technique, and he kindly explained the method to assay about some technique to our graduate student. Today, Hızlan will talk about yeast as a model for cell death, drug, and disease research. So please start.
Okay, thank you, Professor Masaru. Can you see my presentation now?
Yes.
Okay. Today I will talk about yeast model for cell death, drug research, and disease research in the world. So to start, the relation between fungi and human, there's a long story starting from the 6,000 years before Christ and keeping, sorry, it's not going on. There's a problem in the slide presentation, sorry. No, sorry. I think it's okay now. Sorry for the problem. It starts with the Sumerians at first, before Christ, 6,000 years. And then it keeps with the bread production and bakery with cerevisia. No. Sorry for that, but it's not going on the slide presentation. Sorry. Can you see my presentation now?
Yes, I can see.
Okay, I apologize for that. Okay, and it keeps with the antibiotics production in the modern centuries, 20th century, protein supplements, recombinant proteins, and etc. By the Penicillium or Pichia pastoris, and they entered in our daily life fast, very fast. And in the 21st century, nowadays, we use as pombe and as cerevisia cells, also Candida albicans and Pichia pastoris in molecular genetic mechanisms, in cellular biology, DNA repair study. Also nowadays, we talk about that we can use it in cancer study and pharma industry. Yeast models in molecular biology, there are two types of yeast, Saccharomyces cerevisia and Pseudosaccharomyces pombe. In DNA repair study, cell cycle, replication, mutational studies, genome stability, telomere biology also, apoptosis, autophagy, and also we will talk about at the end of our presentation to present our data, preliminary data. And why we choose Cerevisia or Pombe? Because the bidding is, in case of Cerevisia, genome was the first eukaryotic genome to be sequenced in 1996. And this data has opened a new era on cell cycle study, stress response study, cell division, and basic molecular genetics research to understand how all of the molecular mechanisms occurred in our cells. It's freezed. I don't know why. Okay. Cerevisia and Pombe, their genome containing approximately 5,000 and 6,000 genes, and then they can be easily manipulated. So we can monitor the mutation to understand how they can relate to the diseases or something. Similar cell cycle, intracellular signaling and mitochondrial biogenesis, similar programs or controls that we call in the literature, cell death subroutines. There is only intrinsic apoptosis in the yeast, and also we saw the autophagy vacuoles in the Pombe and Cerevisia. They resemble to the chemical and physiological stress response as in the mammalian cells. They have the conserved genes and high level of analogy, and also Warburg and Crabtree effects, which occurred in the reprogrammed energy metabolism in the mammalian cancer cells, which are resembling to the yeast energy metabolism, and there is presenting an experimental opportunity for cancer research. Finally, screen for drug cell and drug phytochemical interactions that we can do in the pharma industry. And there are some examples in the literature, highly cited literature there. And in the genome stability, particularly in telomeric biology, there are so many research in some groups in the world focusing on the telomeric shortening and lengthening events. So by using the Cerevisia and Pombe, we can monitor how the telomeric regulation is done by the more than 80 types of proteins that regulate the telomeric replication. To shed light on telomere lengthening and shortening events, as I said before, Cerevisia and Pombe yeast can be used to understand how the regulation is done by some complexes like RIF1, RIF2, and REP1, or the CST complex, as well as in the evolutionary conserved in the mammalian cells. So they produce active telomerase like the cancer cells. They both are immortalized cells, I mean yeast and the mammalian cancer cell line. So we can anticipate the telomere biology, using telomere biology to understand the mammalian telomere replication. So we can relate the telomeric replication in yeast to mammalian telomere biology. Maybe we can understand and inhibit the cancer progression. And there is an interesting study about lifespan according to the telomere shortening, lengthening events. A group, Romano et al., Romano and his colleagues, done in in yeast. The question was does alcohol increase lifespan or caffeine shortens? Interestingly, caffeine has shortened the telomeric lengthening events in the minus twofold or threefold. In contrast to caffeine, ethanol and other alcohols, but particularly ethanol, can lengthen the telomeric region and its replication 20-fold, more than 20-fold after 300 generations, and it took more than 10 days. So we may say that caffeine can shorten the telomeric replication or telomere lengthening, but it's not real for the. I think it's not real for the ethanol case, because the yeast can produce ethanol also. Maybe they feed the yeast with ethanol to produce its products and metabolites. We don't know. And on the drug research, this figure is coming from a private sector company, and I can't give you the brand name. As you may see in the figure, they use the Pseudosaccharomyces pombe heterozygous deletion mutants to understand its IC50 cytotoxicity results, to monitor its cytotoxicity, and before marketing it, they have to understand and monitor and determine how it can administer it to the people in healthy conditions. So in the third figure, as you may see, we may understand using heterozygous mutant library and which the drug can target which of the genes. So one or a group of genes can be affected by the drug and it's targeted, then we understand then it may survive more than the others, more than the wild types or other mutants. So it means that the X drug or Y drug can target that gene. So we can anticipate that we may use it in the mammalian genome and to monitor it. It will be very useful to understand the targeting and genetic reactions. Contribution to drug research from unicellular to vertebrata. So as I said before, cytotoxicity tests for drug candidates can be performed, and it started, the private sector started to use it. The second one, mechanistic analysis of cytotoxic chemicals causing to apoptosis, autophagy, necroptosis, or mitochondrial disruption. Then those chemicals can be natural products, as we used in our study. There are terpenes, terpenoids, or flavonoids from the plants or from the green or red algae. And generation of mutants, the third thing, to perform functional analysis for the cytotoxic pathway, elucidation of drug response and their gene functions, and the examination of mammalian cells for analogy in the final stage. And our main aim is to understand in the mammalian cancer cells if our natural products worked in the mammalian cancer cells to inhibit them, as well as in yeast, as we monitored in yeast at the first stage of our study. And what we do in our study, what's our aim and what's our vision? Here is an example. We do a mechanistic analysis and perform some cytotoxicity tests. They include mortality and proliferation, using methylene blue assay to see the cells are dead or not, and spot assay and colony forming assay to understand and compare the results between those groups and the control group, and counting with the TOMA under the microscope. And the second phase includes apoptosis. We use the acrid orange ethylenedione bromide dual staining assay to understand the cells are really dead or not. The green cells are accepting the acrid orange dye, and then we may say that they are viable cells. And as you may see in the yellow-orange cells are accepting the ethylenedione bromide if they are dead. So we can understand that they are dead, and this may mark apoptosis or necrosis. We will see in our next slides. To validate our apoptosis analysis, we use the DAPI staining to monitor DNA fragmentation, anucleation, and also we now try COMET assay to validate our DAPI staining assay. But nowadays we have a problem because of the kitting-containing wall is very problematic to take the stain. But I think we will success the COMET assay. The third thing in our study that we have done is to understand loss of mitochondrial transmembrane potential and the oxidative stress if it's induced. And following validations was MBT reduction assay and N-acetylcysteine chemical to inhibit the oxidative stress if they are responsible for the cell death. And the last thing that we have to introduce our study is the autophagy. There are two ways to understand the autophagy has occurred in the cell. One is the GFP dots. If we have the GFP ATG8 fusion protein in our cells, we can monitor under the microscope, fluorescent microscope, the GFP dots and autophagophores. And the second thing to understand and validate our results is the Western blot technique, but now we are working on it because there are some problems in Western blot to understand the cleavage of the ATG8 protein, which is responsible for the autophagy. But we will do it. We will success the Western blot assay, I believe that. And functional analysis for cytotoxicity pathway and elucidation of the drop response gene functions. So what we do in our yeast study, there is a summary about that. We do a spot assay and the colony-forming assay to understand which genes are responsible for cell death or leaving of the cell. And proliferation and mortality assay, including the wild type and also the superoxide dismutase proteins and their mutants also. And at the end, we identify the target genes and which are the potential drug targets, because if they are alive under the pressure of the chemical stress, they should be a potential drug target because they are alive. And if the genes that are mutant in our study can contribute to the cell death, then we can anticipate that they are the real potential drug targets. In our spot assay and colony-forming assays, we can say that. And after that, DCF-DA assay and rhodamine 123 stains to understand mitochondrial transmembrane potential and oxidative stress. And then also we, of course, we published our results in yeast. After that, we wrote another project to support the main aim on the cancer cells, mammalian cancer cells. And at the end of the very end of the study, the xenograph models should contribute to our study, and we can prove our gene, responsible gene, our natural product, and our model, disease model, can work in our way. So in the end, I will talk about our research. We used test organism as POMBE. It's AD, LER, and URA mutant. And we use the natural products or drug candidates, camphor, terpenolen, alpha-2 myrcene, alpha-terpenol, and eugenol, which are the natural terpenes and terpenoids. And then we use them in the S-POMBE cells to see its cytotoxic levels, because they are used in the flowering industry, perfume industry, and all of the phases of the industry which are coming from the factory to our homes, to our offices, to our life. So drug toxicity is another another subject of our study, and finally targeting the cancer drug resistance that I will talk about it in the end of the presentation. We used temozolomide and cisplatin or metformin. They are used for the therapy for the cancer. And then we performed the combined therapy for the eletol, limonen, gentropicosides, phytol, and etc. to monitor they are useful for a combined therapy in cancer cells. Our first molecule was the terpenolen, is a monoterpen. If it induces the apoptotic cell death, and we monitored it induces apoptosis in the increasing concentrations of terpenolen, as you may see in the acridine orange ethylene bromide dual staining assay, and also coloniforming assay and others to validate our results. It induced apoptosis in an increasing way, in an increasing path with the increasing concentrations. And we validate our results by DAPI staining assay. We showed anucleation and the DNA fragmentation in the cell. And oxidative stress was induced by the terpenolen concentration, increasing terpenolen concentration, and there's a dose-dependent oxidative damage in the cell. And we validate it by the, it's not working, sorry. Okay, we validated by the emitter reduction assay. We saw the same results by the DCFTA fluorescence assay. And also we measured gene expression levels of the scavengers like SOD1 and GPX1, and they have been induced by the terpenolen treatment. And these are the measures for the mitochondrial transmembrane potential. And what is the mitochondrial transmembrane potential? The mitochondrial transmembrane potential means that if we inhibit the working of the mitochondria, we can see the decrease in ATP production, which contributes to the cell death, or which starts to cell death, along with the oxidative stress or any other kind of proteins that we haven't know yet. So the mitochondrial transmembrane potential and its measuring is very important in this stage. Then our terpenolen study, after our terpenolen study published in toxicology research journal, we try to understand, and according to our theory, we try to understand terpenolen is a promising anti-cancer agent or not. So we performed experiments using terpenolen and comparing to cisplatin, which is a known, popular breast cancer drug to inhibit it. We compared them. Instead of using the same doses, we used a wide range of terpenolen concentration to understand its IC50 results, because we know that cisplatin's IC50 results. And then there is a tenfold difference between the cisplatin doses and terpenolen doses, but we showed the effect of terpenolen on apoptotic cell death nearly the same. As well as in the Western blot applications, there are some kind of apoptotic markers that we use in our study. One of them is the Box protein, and the second one is the PARP1 cleavage and procaspase expression. In all of the experiments, terpenolen behaved like the cisplatin's effect, and it induces the cancer inhibition in the MCF7 cells, which are the breast cancer cells. And also the HEK cells, human embryonic kidney cells, which are used by our research to understand. It induces the control cells in comparison to breast cancer cells. HEK control cells cannot be killed by the terpenolan treatment in comparison to cisplatin. Also, cisplatin showed some apoptosis induction in the control cells. So, but that does not mean that terpenolan is the best choice for the breast cancer treatment in comparison to cisplatin. In the Annexin 5 PISA, which also we introduced this staining assay to yeast nowadays, and we succeed that. In MCF7 and HEK cells, PI and Annexin 5 assay showed us that the terpenolan also worked as well as the cisplatin. And then you may see the graphs. There's a small difference between the cisplatin effect and terpenolan effect. And these are the oxidative stress levels. Terpenolan can induce oxidative stress in MCF7 cells, which may be related to the apoptosis induction. So, to summarize our terpenolan study proved that yeast can be used in cancer research, at least to understand the cytotoxicity of potential anti-cancer agents. And the leading mechanisms of cytotoxicity are oxidative damage, mitochondrial disruption, and followed by the nuclear fragmentation causing apoptotic cell death. Our second molecule was the camphor, which we focused on it for 1 year. And we monitored its effect on proliferation mortality here. And the first thing that we have to understand was to monitor the oxidative stress is really having an inducing potential for apoptosis. And in wild type cells, DCFTA and MBT results showed us the camphor induces oxidative stress, and it may be related to the cell death. To understand that, we used superoxide dismutase mutants, and in the absence of this scavenger, which is a big scavenger and which is responsible for scavenging and clearing most of those peroxide radicals and hydrogen peroxide and singlet oxygen produced in the cytosol. So if in the absence of SOD1 gene, we can monitor the effect of oxidative stress in cell death clearly. So according to our results, as you may see in the graph in the left side, the scavengers, I mean the oxidative stress, is the most important responsible effect on the apoptosis induction. And these are the arachidone ethylene bromide assay to validate our results and to see the apoptosis induction has occurred by the oxidative stress. Wild type and SOD1 delta cells differ in some kind of different way because the SOD1 mutant cells showed increased apoptotic rates. And we validate our results, of course, by DAPI staining assay to understand the DNA fragmentation has occurred to support our results. And in SOD1 mutant cells, there are an increasing DNA fragmented cells, as you may see in the figure. And then the alpha-tigion, our third molecule, which is a terpen also. It induces the apoptotic cell death in the wild type as Pombe cells, and it's validated by DNA fragmentation, DAPI staining assay, and the apoptotic cells were increasing according to the increasing concentrations of the alpha-2 And of course, we did in the second phase the DCFDA and MBT assay to monitor the oxidative stress is induced. And in the third phase, now we were very sure to see and to monitor the antioxidant effect and oxidative stress effect on the cell death. So we showed SOD1 and SOD2 mutant cells to contribute to the cell death by the oxidative stress, and the SOD1 and SOD2 cells were more vulnerable to oxidative damage. So our theory was proved in that cells with the alpha-2 treatment. And PCA1 and PNU1, there are two kinds of apoptotic proteins. Actually, there are more than the PCA1 and PNU1, but in this study, we used the PCA1 and PNU1 deletion mutant cells to understand if they have a contribution to the cell death here. Then we proved that the PCA1 is seeming responsible for the apoptotic cell death here, but not PNU1. And we have ongoing studies, including alpha-terpineol and also the linalool, which are also terpenes used in the industry. Alpha-terpineol also induced apoptotic cell death here, which is validated by the orange and Duffy staining assays. And in the SOD1 and SOD2 mutant cells, alpha-terpineol was more effective on the cell death. Also, the pro-apoptotic genes, particularly here the PCA1, AF1, and PNU1, did not contribute to the cell death here, as we showed here by the orange, ethylene bromide, and spot assay. So, in contrast to our hypothesis, these three pro-apoptotic genes may have behaved as the survival factors, but we don't know that. According to our results, it seems like that. And it needs the future research to understand how they contribute to survival. Maybe we perform and design the overexpression assays by using plasmids. And the second interesting result of our study is to show the increasing necrosis in the overdose of alpha-terpineol concentration. Tetraconazole, which is known a popular antifungal agent. This is a tetraconazole study to understand azoles are really used to monitor in yeast to understand their effect. And if we can make our hypothesis on the relation between the cancer and the yeast study, then the other azoles that can be used in our study and in our vision. So the tetraconazole also showed apoptotic pathway with showed by the orange, ethylene bromide assay and other apoptosis assays by the PSA. Oxidative stress is also responsible for the cell death effect of tetraconazole here. Also, we measured the mRNA levels of the SOD1 and SOD2, and also GPX1, which are the main responsible proteins in cytosol and mitochondria to clear the reactive oxygen species in the cell. So they are induced. And mitochondrial transmembrane potential was decreased after the tetraconazole treatment. And also we measured some apoptotic gene expressions. The Phoenix one was the best candidate to contribute to the cell death here. Also, linalool oil showed similar results, but there's an interesting figures in here because linalool contributed to cell death. It induces the cell death or inhibit the cellular proliferation. But in DAPI assay, we cannot show the DNA fragmentation, so we couldn't anticipate that it induces the apoptosis. Maybe another path to induce cell death or inhibit cell cycle. Intracellular ROS levels was induced by the second dose of the linalool concentration, and SOD1 and SOD2 cells. These are the preliminary data. I have to say that the preliminary data and results show that the antioxidant proteins can cure the cells in the treatment of linalool. But these are the preliminary data and needs future research and needs replicates here. Then after we cannot see the apoptotic cell death in the linalool treatment, we try to show the autophagy in yeast cells in the treatment of linalool. We used a fusion protein, GFP, fused to ATG8 to give the fluorescence, the green fluorescence, when it targets the autophagophore and start to autophagy. And we may count the GFP dose after cleavage of ATG8 and after docking of the GFP cleavage protein. So as you may see in the results, the positive control was the low glutamate also. I have to say that. The 20 micromolar and 40 micromolar linalool has induced the autophagic vacuoles, acidic vacuoles, by emitting green fluorescent, and we may understand how the linalool induced autophagy or autophagic cell death. In this case, we don't know now yet, but we used the ATG6, ATG8, and other ATG proteins in their mutants. If we succeed to inhibit the autophagy process, to show the autophagy is really responsible for the cell death here. In our preliminary data, we didn't count them. According to our preliminary data, the cells showed decreased apoptotic rates in the autophagy mutants. And we don't know why that, but it seems like the linalool induces autophagy, and this autophagic process can inhibit cellular proliferation along with the maybe a little bit of apoptosis or cell death, but we cannot see the DAPI results, the apoptosis. So maybe the linalool can induce only autophagy, maybe along with the cell cycle inhibition. We will see that. And last thing of our study today, the drug toxicity. We used the temozolomide, which is widely used in the brain cancer curing, particularly glioblastoma treatment, and it interacts with the DNA and causes its methylation. We tried to show the temozolomide toxicity in fusion yeast. Then we can unravel the intracellular mechanism, and we can anticipate that it has a potential to cure the cancer cells. And as we know that it's now used in the glioblastoma treatment, but that is a big problem because the genetic character of every people is different. So now there are individuals that cannot be cured by the temozolomide or cisplatin, which are alkylating agents, because of the drug resistance. There are several ways to contribute to drug resistance in the literature, as we will see in the next slides. So temozolomide and cisplatin, two kinds of alkylating agents, which the first one was used to cure the glioblastoma, and the second one was used to cure the breast cancers. There's a last thing in the temozolomide study that I have to say, that the gene expression of RAD9 gene is induced nearly fourfold in temozolomide treatment. So it really induces apoptosis by the DNA fragmentation, by the damage of the DNA binding of the temozolomide. Then it alkylates the DNA strands and causes the following apoptotic processes by the p53 and p21 and cell cycle arrest and etc. And then apoptosis and cell death were induced. So in the same way, we showed that temozolomide induced cell death in the same way, in the same direction, and within the DNA, within the DNA damage and following pathways. So RAD9 was the first clue of our study. So we want to prove it, the fusion yeast is whether it's used in drug toxicity or the study for the drug candidates, to understand their mechanisms and to anticipate its results, which will be related to the cancer study. The second molecule was cisplatin, as I said before, is another popular alkylating agent in cancer treatments. And cisplatin toxicity demonstrated by our preliminary data, which will be presented in the next slide. The data that we have gained encourages us to upgrade our vision. What is the hypothesis? The yeast can be used as a reliable, reproducible, inexpensive, and faster alternative unicellular model organism in anti-cancer drug research, if our hypothesis is proved by our experiments, and if we can success to relate our study to the cancer cells, cancer cell lines, and also xenograft models, we can prove our hypothesis. So what's our hypothesis? Our hypothesis is cisplatin is an alkylating agent, is transported to the inside of the cell by copper transporters in mammalian cancer cell lines and also in the yeast. It's interesting because there are so many conserved genes, more than 900 genes is highly conserved in yeast, in pombe and cerevisia, in also mammalian cells. So the CTR1 is the first target. The CTR1 can translocate and import cisplatin inside of the cell, and then it's hydroxylated and binds to the DNA itself. And then this alkylating causes the replication inhibition, cell cycle arrest, apoptosis, and cell death in final. And the second thing that the cisplatin affected is the mitochondrial DNA. It can also bind to mitochondrial DNA to damage itself, and it finally causes the mitochondrial ROS production, overproduction here. And then these two kinds of disruption can induce the cell cycle arrest and apoptosis here. These are the two ways of cisplatin's effect. But if DNA repair mechanisms in a cell is working well, or DNA repair mechanisms may be overworking here, in the means of the nucleotide exogenous repair, base exogenous repair, homolog recombination, direct repair, or etc. individually. So one of them is overexpressed here. the effect of the cisplatin can be inhibited. So we call it drug resistance, cancer drug resistance. So the cancer cells can be survived. And so it's stopped. Wow. Sorry. Okay, we were here. Then the cell continues to survive. The cancer cells continue to survive. And why? Sorry for that, but it cannot continue. Okay, the second resistance mechanism can be related to the ATP transporters from inside to outside. They can export the cisplatin molecule to the outside. The ATP 7A, 7B, and multi-drug resistance proteins can do this for supporting the cancer drug resistance mechanism. And another thing to cope with the glutathione, the other thing of cisplatin to pass this blocking, glutathione molecule can bind and scavenge the cisplatin for its elimination. It hydroxylates or conjugates itself to eliminate it. And these are the basic reasons for the drug or multi-drug resistance mechanisms. So what we can do in our study, how we can use the yeast model to understand at first, and the second phase, use our natural products to cure the cells or to enhance the cisplatin or the temozolomide effect on cell death. So there's an example here, one of terpenes. I wrote myrcen, beta-myrcen molecule. If it inhibits the ATP and MRP exporters, it can support the cisplatin's effect on cell death. Inside of the cell, if it can inhibit the glutathione molecule or other antioxidant molecules like SOD1 and glutathione peroxidase and etc., then they contribute to the enhance of the cisplatin concentration inside of the cell. Then it means indirectly supports the cisplatin binding to the DNA. The third thing that the terpenes or flavonoids, we don't know now, can induce the copper transporters expression. If it overexpresses copper transporters and dock on the membrane, so it can help the cisplatin's transport inside of the cell, and then it indirectly affect the cisplatin's binding to the DNA. And the fourth thing of the natural products to support cisplatin's effect is a mitochondrial pathway. If it can induce the mitochondrial DNA damage and reactive oxygen species, so it indirectly can support its contribution to the cell death or cell cycle arrest. And the last thing and the most popular mechanism of the drug resistance nowadays is inhibiting the DNA repair mechanisms. If our natural products can inhibit the DNA repair mechanisms, so it may help to the cisplatin's effect on cell death. So the alkylated DNA cannot be replicated and cannot be transcribed into mRNAs or other RNAs. So the regulation of the cellular proliferation and metabolism are disrupted. Then it means the cell death. So what are our preliminary results about beta-myrcen? As I gave an example for our hypothesis, we did some experiments on Rad9 to understand if it really inhibits the ternary complex, which includes Rad9 and 1,1 complex. So we performed mRNA expression level analysis with cisplatin and temozolomide, and we were sure about it includes the Rad9 inducing. And the second figure, in figure B, cisplatin caused much more cell death in the Rad9 mutants rather than the wild type. And then the colony-forming assay helped us to measure and compare the results between the Rad9 and mutant and the wild type cells. And myrcen alone, in any concentration of myrcen, cannot induce the cell death alone, but cisplatin induced 0.5-fold cell death. But interestingly, when we used myrcen 1 and 2 macromolar doses, combined with the cisplatin, the colony-forming assay showed us the cell death was nearly twofold according to our results in comparison to control group. Then we want to understand myrcen really contributed and helped the cisplatin's effect in apoptosis. In cisplatin 500 macromolar dose group, it induces 20% the cell death, but in the combined therapy group, supported by the myrcen concentrations, in 2 macromolar myrcen treatment with cisplatin induced more than 40% cell death. And our results are validated by the DAPI staining assay to monitor DNA fragmentation, and the myrcen plus cisplatin can induce the cell death much more than the myrcen alone or the cisplatin. And so what is the source of this contribution of myrcen to the cisplatin in the combination? We monitored the mRNA levels of the Rad9 gene. Myrcen alone cannot induce the Rad9 expression, but in the combination treatment, myrcen helped to pull down the cisplatin affected Rad9 levels to the nearly the normal levels. But there's a slightly different decrease in the mRNA concentrations in the Rad9 case. So we may understand, we may solve the drug resistance problems in cancer cells by using yeast cells. Our preliminary data can show us our hypothesis can work in our future study. Then we wrote a project about it, and it's now under review. So we will understand how they can support us or not, if we can show our results and consult them. So to summarize, as Bombay and as Ceravisia are used as unicellular model organisms to understand diseases, to understand the DNA replication, transcription mechanisms, and etc. Maybe all of the cellular metabolism, because they have superior features covering the evolutionary conserved genes and cellular compartments. They are immortal cells, which are bearing the cancer cells in the telomeric biology and forever replication for their chromosomes without shortening events, energy metabolism, organelle function, replication and transcription, cell cycle, control cell death subroutines, including autophagy, the apoptosis or necroptosis, which are making it a valuable model for cancer research and drug cytotoxicity study. Finally, we try to understand how these cells respond to potential drugs and to shed light on the dark side of cell death signals and unravel their genetic background. These are our laboratory members on the left side, molecular cell death laboratory, which are funded under the big laboratory named Life Sciences and Technologies Laboratory in Istanbul University. Our collaborators, Dr. Cenk Kı and Bediye Palabıyık from Istanbul University and our medicine faculty. And also I have to especially thanks to our Yeast Lab founder, Ayşegül Topal Sarıkaya, Dr. Ayşegül Topal Sarıkaya, who is also our vice rector and a big supporter of our results or our study. I especially have to thank her. And our grants is coming from the scientific research projects of Istanbul University and our Turkish Scientific Research Council, Research Authority of Tel Aviv University and Istanbul University scientific research projects. So in the end, I can answer your questions here now, if you have.
Okay, thank you very much for your very interesting talk. Now we can have a discussion or question time. Is there any? First, I will ask some audience if somebody want to ask something. Is there any question? Or I could start first. So you are interested in the camphor and terpene types of compound.
Yes.
So I'm interested in how they induce autophagy. Basically, is there any similar mechanism, or each chemical have a different target to induce apoptosis?
In Pombe or
in Pombe? Pombe. Yeah, in Pombe.
Actually, in Pombe, there is an interesting kind of autophagy evolved, because it seems like the half autophagic pathway in comparison to Cerevisia or the mammalian cells, because you cannot induce using the actinomycin or some chemicals, but we try to understand how our chemicals can induce the autophagy, but we are not sure about that. The autophagy is induced by our chemical treatment, maybe an indirect interaction between them.
So you said the autophagy, the drug which induce autophagy also result cell death in Pombe, and you are using some autophagy mutant. So yeah. I don't remember the name. Linalol.
Yeah.
My question is autophagy mutant more sensitive to linalol or resistant?
I didn't understand your question.
Okay, so
I'm sorry.
You were using ATG7 mutant or ATG some other ATG mutant?
ATG6, 8. Okay.
ATG mutant. So my question is, this ATG mutant is more sensitive to linalool or not?
I think they are not more sensitive. Controversially, maybe they, in wild type cells, they contribute to the cell death by making autophagy.
Okay.
Yeah, because our acrylamide ethylene bromide assay showed us the autophagy mutants. In autophagy mutant cells, there are no more cell death as we figured out there.
Okay, thank you very much.
You're welcome.
Is there is there any question from the audience? Okay, I see the I think Mutaminia Adodian Sam, please ask.
Okay, thank you very much. I'm sorry, I was a little bit, I was on doing some research, my experiment here, so I didn't fully pay attention. But one thing that I catch before about Sensei's presentation, that at the beginning you said that terpenolan is a promising anti-cancer agent, isn't it?
So which one did you say? Linalool, or I couldn't hear you?
Yeah, I think I catch when you explained about the alpha terpineol.
Alpha terpineol, yeah.
Yeah.
It can be can be used as okay.
It can be. I see. I'm just wondering. For example, if we this is for the anti-cancer treatment. So is it? Is it there is a difference between the treatment before the cancer become? As long as I know, the cancer has several levels, isn't it? So
okay, I understand.
Yeah.
So you mean that if we use alpha terpineol as a pre-treatment for the cancer before the cancer progression, so it's not, because of the apoptotic cancer drugs can always induce the cancer progression, because they have some potentials for the inducing mutations, as you know, maybe. So you cannot use alpha terpineol as a prodrug or as a pre-treatment for the cancer.
I see. Okay, it makes sense. Okay, so yeah, just wondering about that. Thank you very much, sir.
Okay, you're welcome.
Is there any other question? I also have a question about Milsen or Misen, you said.
Misen, okay.
That compound is really interesting. So you said that that compound may inhibit a transporter, or
it's our hypothesis.
Hypothesis,
but we don't know whether it is.
Okay, but you found that that compound actually inhibit rAD9 messenger RNA expression.
Yes, in our preliminary data, we showed that, yes.
That is very interesting. Do you have any idea how it happened?
We don't know. Maybe it should be a genetic interactions, because its expression levels are pulled down in the combined therapy. We don't know why. Maybe we understand in our research project that we submitted to our research council if they support us for our study. Now we don't know why.
Okay, but it's very interesting compound. Thank you very much.
You're welcome.
Is there any other question or comments? Okay, Parisa, please ask a question. You should turn on the microphone.
I think there's a problem in the microphone.
Yes, he's
Hello.
Hello.
Sorry, because of the some problem for my device, I used another account. However, my question is about the actually the type difference between the human or actually mammalian cell and yeast, because for yeast we have a single cell, we have a separated cell, and in case of the mammalians we have a connected cell. Am I right? It's something like this. I don't know if we can completely say all the data could be usable for the, for example, mammalian cell or not, or actually also yes. My question is about it.
Okay, that's a good question. Thank you very much for that. There are so many conflicts about our yeast study coming from the cancer researchers, because they don't want to, maybe they don't want to make a relation between the yeast and cancer cells, and they have some right questions, and in the other side we have the right questions. Our questions are why the evolutionary conserved genes, much more than 2,500 genes are conserved, and in the orthologs genes we have the 900 orthologs genes in mammals and the yeast. This is the genetic case that I have to say. And the other case is much more resembling to the mammalian cells. I mean the cellular metabolism. There's a mitochondria which is completely resembling to the mammalian cells mitochondria. There are some other kind of organelles and endoplasmic reticulum, the nuclear metabolism, and as we showed, the cisplatin and temozolomide and other agents act as they are the cancer cells because they killed our yeast cells in some ways, which are the same in the expressed in the mammalian cells. So I know there's a conflict between the yeast researchers who are making the yeast cells a cancer model, and mammalian cancer researchers. So we will see if it can be used as the cancer model or the model for anti-cancer drug potential of yeast. We will see.
Thank you. Therefore, did you check the normal cell with the drug? I mean, that you check the mammalian cell in case of the terpinol, or am I right? Something like this you explained about the kidney cell, I don't remember, and breast cancer cell. Am I right? You did some experiment. Did you check for the normal cell also, or did you see the cells in normal cell also, or not?
Okay, good question. Thank you. We used the normal cell, human embryonic kidney, 293 cells as a control cells in comparison to MCF7 breast cancer ER positive cells. And we showed that the terpinolene induces much less cytotoxicity against HEC control cells in comparison to cisplatin.
Thank you. Thank you.
You're welcome.
Thank you for me.
Welcome.
Okay, so is there any other question? If not, I would like to close this seminar by thanking Hızlan again. Okay, thank you very much. It was very nice talk.
Thank you.
And
Greetings from Turkey.
I want to take a picture with you. So after the seminar, so could you please stay a little bit longer, 2 or 3 minutes?
Okay. I'm here.
Okay. So I want to take a picture. Okay. So thank you for attending the audience. The seminar will be closed now. And I just want to take a picture with you. How I don't know how to do that. Maybe I can spotlight.
Maybe we can use the phone, the cell phone.
Yeah, spotlight. Maybe you and I can use a spotlight. I don't know how. Okay, so you can also spotlight.
Maybe we can take a screenshot.
Yeah. Okay, so yeah, we can take a. Maybe I should do turn on the.
No, I cannot see your camera. I cannot see your figure.
Really?
Yeah, really.
Okay. So anyway, I will take your picture first, and then.
Okay. Okay. I can see you now.
Okay. And I wanted to show the picture you and me side by side, but maybe I can modify later.
Okay.
Okay.
If you record our presentation and of the discussion, can you send me the link of the iCloud or something?
Of course. Yes.
Thank you very much.
Yes. And it was very nice talk, and I have many other questions, but maybe it was very long, so we could continue the discussion. Maybe keep in touch.
Okay. Maybe you will come to Turkey in the future, and
yeah. Of course,
you are welcome everywhere.
Yes, thank you very much. I'm really interested in visiting Turkey.
Okay.
Or you could also visit Japan in the future.
I will plan to visit Japan and your university in the future.
Yeah.
Maybe after the pandemic,
Yes. Yeah.
Okay.
Thank you very much. See you.
You're welcome, and thank you. And greetings from Turkey to Japan.
You too. Bye-bye.
Goodbye. Bye.